today it's a very good opportunity for me just to show a little bit more progress in our metabolomics analysis of the patient samples from our study. And basically we focus more on the inborn errors of metabolism. And yeah, we talk about this scientist, Dr. Gar, and he basically gave this very famous Cronin lectures in front of physicians talking about his discovery of a new disease called acaptonuria, also called black urine disorder. And he also for the first time linked the genetics to the inborn errors of metabolism. He found that actually this disease is caused by the autosomal recessive mutation of a gene called HGD. And that gene is involved in the catabolism of tryptophan, not tryptophan, tyrosine and the phenylalanine pathway. And so I think his lectures basically are recognized as the foundation of current modern medical geneticists. I think 100 years ago, there were probably only like a handful of IEMs discovered, but now after 100 years, and we have hundreds of IEMs identified. Also, a lot of patients with the metabolic phenotypes, but with undiagnosed situation, especially known disease genetic causes were identified. So to understand more about the IEMs, especially undiagnosed IEMs, let me how to press that. Oh, I cannot move the slides. Click it. Oh yeah, good. So in 2014, Dr. Ralph DeBardinis, and who is also our geneticist in Children's Medical Center and UT Southwestern, initiated this program called Genetic and Metabolic Disease Program. We call GMDP. To be short. And so this is a very unique combination of the clinical team and also our research lab. And we aim to understand more about our human biology and metabolism using the disease models, and also, trying to provide more insights to improve the lives of children, especially for their disease diagnosis and treatment. And we basically recruit patients from the Division of Pediatric Genetics in Children's Medical Center. And we also have a physician work on the adult genetics clinic in UT Southwestern to recruit the patients, both with the diagnosed or undiagnosed situation, and also their families. Yeah, in the lab, and our lab is mainly located in the North Campus of UT Southwestern Children's Research Institute. And we have quite a lot of platforms and to analyze the patient samples and build different experimental models to understand more. If you are interested, you can check our website. It's passed. Yeah. So here I give you a very brief introduction of our clinical research program study, especially the workflow. And basically, we have our patients recruited in different clinics, as well as our collaborators. We have very important collaborator in Pakistan, the Aga Khan University Hospital. And we have those patients, including both the families, patients, and also some healthy donors. And so we collect their whole blood and also plasma. And for the patients with the undiagnosed disease, and we are trying to piggyback their skin biopsy to derive the fibroblasts as well. And our main analytical platforms, including the whole exome sequencing, and we prefer to sequence the whole family because that's going to be very helpful to understand the inheritance of the disease. And we also established the metabolomics platforms, and including targeted and non-targeted as well. And we focus more on the targeted metabolomics currently, and to identify the abnormal metabolic profiles in the patients. And we also use the patient samples, patient cells for some functional assays, and it depends on the gene. So using this very unique platform, and we can generate a lot of interesting data. And basically, our physicians are particularly interested in any information that can be used for clinical diagnosis and all indication of the treatment. And in the lab research, and we have trainees, we have scientists working on different cell models, animal models, and including the in-depth analysis of different clinical samples. And currently, in the past few years, we already published several cases. If you are interested, you can check out our papers. And I think these are very important and precious resources for our basic research on IEMs. And to take advantage of the big data sets generated from this platform, and our bioinformatics expert, Lynn, also make all the data available in our web-based data portal. We call it GMDP data portal. And she developed embedded tools to visualize the data and analyze the data, and as well as there are a lot of bioinformatics analysis we can use. And this website is going to be public when our paper get published. And in this big cohort data sets, currently we already have like over 200 patients, including their families, get a whole exome sequenced. And we have run metabolomics analysis using both patients' plasma and their fibroblasts, and trying to build this data set. And so next step is how does a cohort data set look like? So this is our bioinformatics expert, Dr. Lin Chai, and she using her portal, and we can visualize the data. And before that, we are very interested in to see how the whole data set look like. So here is the summary of our cohort subjects, and we have this many patients recruited from the U.S. clinics, and we'll have this many patients, about almost 200 patients recruited from the Aga University Hospital, Pakistan, and also, for the majority of them, we have their both whole blood and the plasma samples for all the different kinds of analysis. So one example I would like to share with you is using this data set, you can do a lot of correlation analysis, age, sex, and also their the families and ethnicity like that. So this is one example for the age, and we basically generate this kind of plot to visualize, what's the age distribution across different cohorts. And you all see point here, pro-band we call the affected children and all the patient. And the Pakistani cohort, all those are younger kids. They are younger than 18 years old. And for the US cohort, we do recruit some adult patients as well. Yeah, you all see here, there are some dots spread up above. And one interesting finding we have is we see the plasma creatinine increase with age. I think this is quite interesting and actually has been reported a long time ago. So creatine is a waste product of muscle degradation, right? And blood creatine levels have been used to evaluate the renal function. In human body, in healthy human body, kidney can filter the blood creatinine and excrete it through the urine. And with the age increase, and you all see the clearance of creatine from the blood kind of reduced. So this is one example showing here, and you all see the increased creatinine in the plasma with age goes up. Yeah. So we also perform other kind of dissimilar correlation analysis. Definitely you can see a list of metabolites that kind of correlated with different changes at different groups. So this is one aspect that we haven't, quite finished the analysis and the data interpretation yet. This is one example. So the next question we ask is how to use the data. And we have quite a lot of patients with different types of IEMs, and what we can do, use this data to answer different questions. And Danny Vu, and he's our mass spec specialist, and he basically ran all the plasma and the fibroblast samples on the Agilent QTOF machine, and our data generated from that screen panel. So this is one example, glutaric aciduria blocked type 1 disease. So this disease is a very rare organic acid disorders. It's caused by the inherited deficiency on the gene highlighted here in red is GCDH. It's a glutaric-CoA dehydrogenase. And this enzyme is involved in the lysine degradation pathway, you all see here. If this gene is mutated, loss of function, and glutaric acid basically is a biochemical marker for this disease, kind of increase in patients. And this is a snapshot of our cohort of our web tour analysis result, and you all see those three. Actually, those are two dots. Those three outliers are all GA1 patients, And here, it jumps up here. You will see multiple different intermediate metabolites along this pathway, including the hydroxy kinurate downstream of the tryptophan metabolism also fit into the same pathway are accumulated. So basically, we think metabolomics data can give us a broader view of the metabolic abnormalities in specific patient samples. Yeah, I think this is a kind of, makes sense to us. And in addition to those metabolites shown here, and definitely we also see other metabolic pathways affected. But currently we need to understand how those pathways are affected directly or indirectly due to the GCDH mutation in this case. So the next example is I want to show you how we use the metabolic findings to direct the therapy. So this patient is a 29-year-old female. Previously, she was healthy, and she was recruited by our Dr. Marky Magnan, and he's a geneticist in adult genetics clinic at UT Southwestern. And when Dr. Magnan saw this patient, actually, she was just to deliver her first baby, and her blood ammonia level is very, very high, hypoammonemia. And this patient at that moment developed very complex symptoms, including acute confusion ataxia, and also very severe symptoms, hemolytic anemia. So extensive clinical tests shows the patient has defects in urea cycle disorder, but the clinical sequencing tests cannot figure out the exact genetic course. So we collected this patient's blood as well as her parents' blood, and for a very quick whole exome sequencing. And we found a mutation, 2 mutations. We call it Hong Kong heterozygous, which means one is from mom, one is from dad, 2 different mutations. And in this gene, N-acetylglutamate synthetase. So one mutation is a missense mutation, and very easily to be detected by the clinical lab as well. Another one happened at the splicing site. So basically, this one kind of failed to be reported by the CLIA lab, but we found this one. So NAGA is a very essential enzyme in the first step of urea cycle. So basically, this enzyme can catalyze the synthesis of the cofactor for CPS1. CPS1 is right here, the first step enzyme to bring the ammonia to this cycle. And the most important cofactor called NAG, it can activate CPS1 activity in the urea cycle. If you lost the next function, the whole cycle shut down. That can cause very severe symptom in the patient. And many of those genes in this cycle has been linked to the different types of urea cycle disorders. So fortunately, there's a drug called carbaglu, also the carglumic acid available. It's an analog of the NAG. So here is the NAG, the methyl group here, replaced as an amine group here. So this is the carbaglu. So carbaglu is also functionally equivalent to the NAG. It can kind of, supplement, you can supplement the patient with the drug, and then it can activate the first step of urea cycle. So we give this patient a lot of pills every day, just to take a lot of pills. And so here we measure the glutamate and the glutamine levels, and her before the treatment, her level is very, very high. You will see the pre-treatment, and then after treatment, 3 days and 1 month, we see the normalization of those 2 amino acids. And also, this patient has been taking this drug for over 4 years, and during these 4 years, he keep taking it, and he has 2 new babies, and the hypoammonemia symptoms never occur during this process. So it's kind of very, very, very good story. The next step, and we ask our question, the question we ask is how we use our metabolic information to discover new undiagnosed IEMs. So here is an example, and this one show you the level of 2-hydroxyglutarate in patients' plasma samples. So we have this many patients here, and there are like three different subjects with very high level of 2-HG. And this patient 1, patient 2, and patient 3. And patient 1 is a female, and with severe lactic acidosis, a very high lactate level, epilepsy, and development delay. And the patient 2 and 3, they are from a family out of Pakistan. They are siblings. both of them have the intellectual impairment and ataxia. So this high level kind of, very interesting. We don't know whether they are same disease or different. So the first one is patient 1, and her blood lactate level actually was consistently high across many years. And you will see, this is the highest one. Oh, this is the highest one. And when she had the metabolic decompensation, and our exome sequencing analysis of this family found out this patient has a mutation on the gene called the LIPT1, and also like 3 out of those 5 girls in this family affected. Those 2 kind of, disease are kind of very early. We can get their samples to validate the genotype, but this patient has 2 mutations inherited from parents. So LIPT1 is a very important enzyme involved in the modification of essential 4-keto acid dehydrogenase, including pyruvate dehydrogenase, ULC-PDH. This is a very important one for the glycolysis, link glycolysis to TCA cycle, sorry. And all those 4 enzymes are involved in the TCA cycle related to the mitochondrial TCA cycle function. And especially the PDH, I think this is one very important to generate acetyl-CoA and then feed into the TCA cycle. So this enzyme is very important function. And what we found in this patient is when we run her plasma samples together with all other controls, including the other healthy sibling, you will see very distinct pattern around here. And we see the 2-HG level is very high. We're trying to find out which enantiomer is high, and we found both. This is a combined elevation of L form and D form of 2-HG in this patient. And also, as I say, PDH provides acetyl-CoA to TCA cycle. Acetyl-CoA is also important for lipid synthesis. And we also run the patient's plasma samples using the lipidomics method. And we found that the membrane proteins here, including PCP, ASM, and ceramide, they are very low in this patient. But the tag, the triacylglycerol, and kind of very high. For here, we think that might be our kind of indication of the dietary intake. So basically, you all see the patient's plasma samples give us quite different metabolic profiles. Yeah, so we also get the patient's fibroblast. And here, when we culture the cells in conditional medium, basically nutrient-restricted medium, you all see they grow very slowly. If we kind of express back the white type LIPT1, and you will see the cells kind of much healthy, look here. And we use the fibroblasts to do the metabolomics analysis as well, just to confirm and validate the metabolic deficiency we observed in the plasma. And the top ranked pathway is TCA cycle. And here, just an illustration of all the changes in the metabolites in TCA cycle and the related pathways. So I think this is a very important platform and also strategy for us to have a broader view and more in-depth phenotype, metabolic phenotyping of individual IEM case. So the last case I want to share with you is how we integrate the metabolomics, genomics, as well as our functional assays to facilitate IEM detection. So if you remember, there are like 2 other patients showing very high 2-HG level, and those are those 2 siblings, and they have the homozygous nonsense mutation on a gene called L2 hydroxyglutarate dehydrogenase. And this enzyme basically can convert L2-HG to alpha-ketoglutarate, but the enzyme is dysfunction, and definitely you're going to see the accumulated R2-HG. So this is their plasma data, and you all see those patient 1, patient 2, they are outliers, right? And with very high 2-HG level, parents are normal, even though parents are carriers. And we also obtained their fibroblast, and the significant outlier is 2-HG as well. So this is kind of very consistent between the plasma and the fibroblast metabolomics profile. And so because those patients has the defects in neurological system, they are intellectual delay and they have ataxia, and how to model this system. And definitely we can generate a mouse model to do it. And Wen Gu, very talented postdoc fellow in our lab, and he used the patient fibroblast to reprogram to the induced proprietary stem cell. And those cells can be differentiated into neuron progenitor cell and then into the mature neuron cells. So here are the established this system very well characterized all the markers and the stages of the differentiation. And so using this one, and he basically differentiate the control and as well as the patient cells into neuron and to compare their different neurological features. So one example I want to show you is very exciting here is you all see this is a patient one's neuron cells derived from her fibroblast and then iPS cells. And it looks like this. It's not very beautiful. It's not very typical neuronal cell network like that. And then he used a CRISPR-Cas9 technique to correct that mutative site. If you remember, that's a nonsense mutation, just generate a stop codon, and then correct that site to the Y type. And then when we look at the morphology, you see the beautiful neuronal network here, and all the tubulin staining indicated the neurite kind of stretch out. And when also measured the neurite length, and he found after correction, the neuron length kind of extended, increased, kind of close to the normal situation. So this kind of normalization and is a very, very important functional indication about how L2HGDH mutation caused the effect in neuronal differentiation. A very nice model. So for summary, and currently we focus more on the cases that we have both plasma and the fibroblast metabolomics, because that can give you different aspects in terms of the metabolic interruptions in the genetic situation. So and we definitely compare whether you can see the overlapping metabolites and between the plasma and the cells or not. Yeah, using those information, we can validate the disease associated biochemical markers and characterize the affected metabolic pathways, like Stu showed you, and they also did a kind of pathway enrichment analysis. And we can definitely use this data set to define new metabolic features. But if you remember, IEMs are very rare, and we barely have like over 2 or 3 or 4 patients with very similar gene mutations or diseases in our cohort, except those very common ones. So in that way, we're trying to divide our cases in different scenarios. And the first one is we have some consistent findings in plasma and the fibroblast, and those are kind of make us more confident about the findings of metabolic outliers. But sometimes we do see very different abnormalities in plasma and the fibroblast, or even opposite. One metabolite high in plasma, another one the same metabolite is low in the fibroblast. So that's a kind of very interesting situation. We're trying to do more analysis. And the third scenario we are having is we have like the diseases, different diseases, but the causative genes are linked to the same pathway. And so that's a very interesting situation. We want to figure out what's the common features, what are different features regarding to different gene mutations. So the last one, definitely, we want to characterize the previous unknown IEMs. There are a few cases we are working on right now. So, and I'm happy to stop here. And at last, I want to acknowledge this kind of, very big team. And at the top is our debriefing lab. We have a big team to work on different aspects of the project. And also the bottom one, I would like to thank our clinicians and especially genetic counselors, first of all, genetic counselors. And we have co-workers and we have dietitians working this team. And also Dr. McNa is our adult clinic geneticist, and Dr. Lin Chai is our bioinformatic expert. So specifically, I would thank our CRI metabolomics core. Those guys are fantastic, and they can work no stop. Yeah, never stop the machine. Yeah, to analyze, because we have like 700 samples need to run, and it takes like about a month to finish the run, and it takes another 2 months to finish the peak calling like that way. Yeah, so they did a fantastic job. And I would also like to thank our collaborator. This is a very short list. We have a other collaborators and to send us samples and to discuss different scientific questions with us. And we do learn a lot, yeah. And I would like to thank all the CRI labs and particularly genetic counselors. And they are always, underappreciated in all the presentations, but they are the person who truly, help us to recruit all the, a few hundreds of patients and their families. Yeah, and lots of patients and families are very supportive to our study. And last, we received big support from Howard Hughes Medical Institute and as well as Moody Foundation. Yeah, okay, I'm stop here. And any question?